Myself Vinita Tiwari and welcome to my YouTube channel Learn Life Science. So this video is going to be detailed introduction about phytohormones gibberellin. Okay, so we all know phytohormone are the organic compounds that are involved in a proper growth and development of any plant. Okay, but when we talk about gibberellins, it acts specifically in the initial stages of plant growth. Okay, for example, it is involved primarily in the plant height as well as seed germination. Okay, so we are going to deal with in detail concepts regarding gibberellin, precursor synthesis as well as the gibberellin synthesis, the compartmentalization of the site of synthesis. Okay, now let's get started. So gibberellins were initially discovered in Japan, where the farmers noticed that their seedlings or their rice plants were getting infected by any kind of fungus. Okay, so Japanese scientists discovered that a fungus named Gibberella fujikurai was a pathogenic fungus that was responsible for the infection to the rice plants, and the disease was commonly called as the foolish seedling or a Bacone disease. Okay, so before other British or American scientists, Japanese scientists were the first who discovered this phytohormone gibberellins. Okay, now in recent day, today more than 130 gibberellins have been identified in plants, in fungi as well as in bacteria also. Although many few of them is having a biological activity. Okay, and those who are non-bioactive, they generally act as a precursor for the bioactive forms. Also, they are responsible for the deactivation of many metabolites. Okay, the major bioactive gibberellins includes GA1, 3, 4 and 7. Okay, so you can imagine in such a diverse group, that is in such so many gibberellins, only 4 gibberellins are biologically active. Only 4 gibberellins are involved in a proper biological activity functions. Okay, now what is the structural nature of gibberellins? So gibberellins generally belongs to a group of secondary metabolites which we commonly called as terpenes or terpenoids. Okay, so gibberellins are derived from the terpenoids. So here are some of the important functions: that they are having the capacity to stimulate hyperelongation of intact stem, because when they were identified, when they were observed the disease of rice seedlings, the rice seedlings were having the maximum height as compared to the normal plant growth. Okay, so they are generally involved in increasing the stem height. The second prominent feature is it is involved in the seed germination and mobilization of endosperm reserves during the early embryo growth. Okay, as well as leaf expansion, pollen maturation, flowering and fruit development. Okay, in the next video tutorial, I am also going to give in detail about the gibberellin signaling pathway as well as the endosperm functioning. Okay, now moving next. So let's talk about the structure, its precursors. Okay, so all gibberellins are generally diterpenes because they are composed of 20 carbon unit and each having this common end gibberellin structure. The end generally means that they are the enantiomers. Okay, they are having the mirror image structures. So you can see here its structure. Now every gibberellin have this basic skeleton, but 4 important characteristics differentiate biological from non-biological activity gibberellins. Okay, for example, a biologically active gibberellin will have a carboxyl group at carbon 7. Okay, then carbon 19 gibberellins are more biologically active than carbon 20 gibberellins. Okay, in addition, gibberellins with 3 beta hydroxylation, 3 beta 13 dihydroxylation or 1,2 unsaturation are more active. And lastly, those with both 3 beta hydroxylation and 1,2 unsaturation exhibit highest activity. Okay, so these are some of the characteristics that differentiate biological from non-biological gibberellins. Okay, now moving next, there are generally 3 principal sites for the synthesis of gibberellins. As I have already told you, gibberellins are generally involved in the initial stage of the plant development. Okay, you can see here they are present in the developing seeds and fruits, the young leaves of developing apical buds and elongating shoot, as well as the apical regions of the roots. Okay, apical means at the tip. Okay, and lastly, you can see immature seeds and fruits are the prominent sites of gibberellin biosynthesis. Okay, and that's why I was saying that gibberellins play a very crucial role in the initial stage of plant seed germination to initial plant growth and development. Okay, so you should remember this point. Now let's talk about the precursors of gibberellins. So gibberellins are terpenes sharing a core pathway with several other hormones and a wide range of secondary product. As I have told you, it is derived from secondary metabolite terpenes. They are diverse and complex. Okay, secondary metabolites that are belonging in group terpenes, they are generally diverse due to its cyclic structure as well as many conjugated form. Okay, not only gibberellins but many other compounds like abscisic acid, brasinosteroid, cartinopigment, as well as sterol and latex are also formed or derived from these terpenes. Okay, and therefore the 5-carbon isoprene unit is the basic building block of terpenes and terpene derivative. You can see here these 5-carbon isoprene unit is the building block of terpene. Okay, and if it is the building block of terpene, that means it will be also going to be the building block for the terpene derivatives, that is these compound mentioned. Okay, when we talk about the biological isoprene unit, we generally use it in the isopentenyl diphosphate form. Okay, when two phosphate group are attached to this isoprene unit, they are biologically involved in the gibberellins or other compound synthesis. Okay, now let's talk about its synthesis. So isopentenyl diphosphate is a product of two different biosynthetic pathway. Okay, one located in the cytoplasm and one located in the chloroplast. Let's see both. Okay, so these are the two pathways mentioned for the synthesis of isopentenyl diphosphate. Okay, so in the first pathway you can see that is the cytoplasmic pathway. 3 acetyl-CoA molecules generally condense together to form hydroxymethyl glutaryl-CoA. Okay, and the most important intermediate, which is a committing step, is the mevalonic acid synthesis. Okay, and therefore we also called it as a mevalonic acid pathway. And now after addition of 2-phosphate by ATP, it forms mevalonic acid pyrophosphate, and finally there is synthesis of isopentenyl pyrophosphate. Okay, whenever we talk about cytoplasmic isopentenyl pyrophosphate synthesis, the most important intermediate or the committing intermediate for isopentenyl synthesis is the mevalonic acid. Okay, isopentenyl diphosphate or pyrophosphate can undergo isomerization into another form, that is dimethyl allyl pyrophosphate. Okay, now let's talk about the synthesis in chloroplast. Okay, here glyceraldehyde 3-phosphate combines with a pyruvate molecule. Okay, glyceraldehyde is generally a 3-carbon compound as well as pyruvate is 2-carbon compound. Both combine and there is a release of carbon dioxide and therefore they form deoxycellulose 5-phosphate. Okay, now on addition of NADPH, you can see here the most important intermediate in this pathway is methyl erythritol 4-phosphate. Okay, it is undertaking the committing step. Okay, and after addition of 3-phosphate molecules, we get our desired product, that is isopentenyl pyrophosphate, which can also undergo isomerization into dimethyl allyl pyrophosphate. Okay, so this was all about the two pathways for the synthesis of precursors of gibberellin. Okay, and those precursors are isopentenyl diphosphate and or you can say it dimethyl allyl pyrophosphate. Okay, and the locations are cytoplasmic and chloroplast pathway. Okay, location. Now as we have learned about the synthesis of IPP, Now we will switch to another compounds that are also compartmentalized for their synthesis. Okay, for example, in cytosol, you can see here isopentenyl pyrophosphate is acting as a main compound for the synthesis of cytokinins, then sesquiterpenes, phytosterol as well as brasinosteroids. Okay, you can see here the main intermediate that is acting upon in this pathway is the farnesyl pyrophosphate, okay, which is acting as a precursor for sesquiterpene, which is also a terpene, and a phytosterol which is involved in synthesis of brasinosteroid. Okay, similarly, chloroplast is also involved in synthesis of many important metabolites. Okay, in chloroplast, the IPP formed is involved in the synthesis of monoterpenes by using intermediate geranyl pyrophosphate. Okay, geranyl pyrophosphate, a 20 carbon compound, is involved in the synthesis of gibberellins, carotenoid, a pigment, abscisic acid, another phytohormone, plastoquinone that is used in photosynthesis, as well as the phytotail of a chlorophyll molecule. Okay, I think you should note this all. Now, moving next, talking about gibberellin synthesis. Till now, I have told you about the synthesis of gibberellin precursor, that is indole isopentenyl pyrophosphate in cytoplasm as well as in chloroplast. We have also seen different molecules that are produced through isopentenyl pyrophosphate in cytoplasm as well as chloroplast. Now moving to pathway. Now we will learn about the synthesis of gibberellins directly. Okay, so let's get started. So firstly, we have seen that now we are having the main product, that is isopentenyl pyrophosphate, which undergoes isomerization to dimethyl allyl diphosphate. Okay, so the synthesis of gibberellins generally begins with the C20 isoprenared geranyl pyrophosphate, okay, which we also called as GGPP. As you can see here, IPP on addition of another IPP group, that is now it becomes 10 carbon, it forms geranyl diphosphate. Okay, so geranyl diphosphate is produced from 2 IPP molecules of 5 carbon, and therefore it is now a 10 carbon compound. On further addition of IPP, it forms farnesyl diphosphate, which is 15 carbon compound. On again addition of IPP, there will be production of geranyl diphosphate, which is a 20 carbon compound, and all these reactions are taking place in the plastid. Okay, so it is also localized. Now the reaction is the cyclization of the molecule. Okay, now the two reactions involved, you can see here, are involved in the cyclization of the structure. Okay, so the cyclization process, for example, geranyl diphosphate is getting converted into copalyl diphosphate, which further gets converted to end corine. Okay, so this both is involved in the cyclization of structures, but both these steps are inhibited by some molecules which we also called as dwarfing agents or anti-gibberellins. As mentioned here, it is AMO1618 cyclosyl phosphorn D. Okay, and therefore these dwarfing agents can lead to dwarfism in plants. Okay, now moving next. Following cyclization, the carbon at 19 position of corine molecule undergoes 3 successive oxidation. Okay, so now we are talking about this step that is undertaking in the endoplasmic reticulum. The carbon 19 position of end corine undergoes 3 successive oxidation, as you can see in the reaction, to form corinoic acid. Okay, and this pathway is inhibited by another dwarfing agent, that is enzimidole. Okay, now the oxidation of corine to corinoic acid is inhibited by enzimidole, another dwarfing agent, as I have told you. Now at last, the final step involves hydroxylation at carbon 7 and contraction of the B ring to form G12-7 aldehyde. Okay, now corinoic acid is converted to one of the inactive gibberellins, one of the first gibberellins that is synthesized in this pathway by taking 2 steps. The first one is the hydroxylation of carbon second. The second thing is the contraction of the B ring to attach a aldehyde group. Okay, now let's see it through a structure. So okay, so let's see. So I was talking about this precursor hydroxycorenoic acid getting converted into Ga12 aldehyde. Okay, so you can see here this is the precursor corenoic acid. The first thing is here is the hydroxylation. As you can see here, the addition of OH at carbon second, and the second thing is the contraction of the B ring. From 6-membered ring, it is getting converted to 5-membered ring. It is clearly mentioned here that end corenoic acid is getting converted to aldehyde by taking these two main steps. Okay, so this is very important in this point. Okay, now the G12 aldehyde is getting converted to G12. For this, it is important that there will be oxidation of aldehyde. There will be oxidation of aldehyde group on carboxyl 7 to a carboxyl group that will give Ga12. You can see here the aldehyde group CHO. Okay, to convert Ga12 aldehyde to Ga12, we will simply have to remove this aldehyde group and convert and add here a carboxyl group. Okay, so it is a really simple pathway. You can see here the enzyme involved. This S represents the synthase, KO represents corine oxidase, KAO corenoic acid oxidase. Okay, now let's give it a more detailed view. Okay, now we are aimed for the synthesis of the active gibberellins. Okay, so till now we are having this Ga12. Okay, Ga12 aldehyde get converted to Ga12 by removal of the aldehyde group. Okay, now when you will use Ga2 oxidase, means 2 oxidase if you use, then it will get converted to Ga110 form. Okay, the enzyme involved here is Ga2 oxidase. If you will use Ga13 oxidase, it will convert Ga12 into Ga53. Okay, and similarly I have named all such enzymes that are involved in their respective conversions. Okay, and you can also see here that one enzyme is undertaking so many steps. Okay, let's see one by one. You can see here Ga12 is getting converted into Ga15 by 20 oxidase. Similarly, it is converting into Ga24, and now the main important step, that is conversion of Ga24 to Ga9 by the same enzyme 20 oxidase. Okay, the thing to remember here is that Ga9 is getting converted to active gibberellin Ga4 by the enzyme 3 oxidase. Similarly, Ga20 is getting converted to active gibberellin Ga1 by the 3 oxidase enzyme. You can also see here Ga5, a gibberellin, is getting converted to active Ga3 by 3 oxidase. Okay, so you can clearly get here is that Ga3 oxidase is the enzyme that is involved in the conversion of inactive gibberellins to the active form. Okay, now these active gibberellins can also get inactivated by the these active gibberellins can also get inactivated by the biologically inactive gibberellins. Okay, we are also going to see that. For now, just see the production of the 4 biologically active gibberellins, that is 1, 4, 3 and 7. Okay, you can see here and memorize it easily that the R group attached here, when added with hydrogen group, then it forms Ga9. Similarly, if OH group is attached, then it forms Ga20. Now, by using Ga3 oxidase, the main enzyme involved in the synthesis of biologically active gibberellins, you will see here there will be production of 1, 3, 4, 7 with their respective R groups in the structure. Okay, I hope it is now easy for you to understand. Okay, if you still have any kind of doubt, you can add in the comment section. Now let's see how the active gibberellins are getting converted in inactive form. Okay, So just look out right here that GA1 is biologically active, okay, and when its concentration is maximum in a cell, 2-oxidase will convert it into GA8 form, that is inactive form, okay. Similarly, GA4 will be inactivated by 2-oxidase to GA34, which means that GA2 oxidase is an enzyme that is involved in inactivating the biologically active gibberellins to biologically inactive forms, okay, whereas in activation we have seen that the main role is played by 3-oxidase, okay. So I hope now you understood it more well. So this was all about the synthesis of gibberellins. If you are having extra points to add on it, you can please add it to the comment section. Now we are going to talk about the stem growth, the most important function of gibberellin. Okay. So in which enzyme is generally involved in elongation of stem height? Okay, so if a gene is involved in the synthesis of an enzyme, we all know that there will be a wild type as well as a mutant type, okay, mutant alleles more specifically. Alleles are generally the two different forms of a gene present at the same loci, okay. So GA1 is biologically active gibberellin controlling the stem growth. Now again, it's an important point that which gibberellin is involved in controlling the stem growth. It is the GA1. Now, tall stem do contain more bioactive gibberellin than dwarf stem, and that the level of endogenous bioactive gibberellin mediate control of tallness. It is a direct statement. Second thing, the gibberellins of tall plant containing homozygous LE allele, you can say here it is designated in capital letter, which is also the wild type, were compared with the dwarf plant having the same genetic makeup except the LE mutant allele. This sentence means that the wild type is the capital LE allele, whereas the mutant is the small le allele, okay, responsible for dwarf plant, as well as the wild type is having the normal tall growth. Now, capital LE and small le are the two alleles of the gene that regulate tallness in peas, okay. Now we all know alleles are present in pairs in any living organism, okay, in any plant or in any human. Whenever there will be presence of capital LE, the plant will be obviously normal tall. If the case is recessive homozygous, that is small le, the plant will be dwarf, okay. And if the condition is heterozygous, the plant will be little more than tall, that is little more than normal, that is due to this the dominant nature of the capital LE, okay. I hope you understood it. Now moving on next, as we have seen that the precursor of biologically active GA1 is GA20, okay, and the main enzyme that is acting upon here is GA3 beta hydroxylase, okay. And now here I have mentioned a point, that is precursor of GA1 is GA20. If GA20 is applied to homozygous dwarf plant, they fail to respond, although they respond to applied G1, okay. What mentioned here is that take a dwarf plant, okay, and give it application of GA20. The result will be that it will be failed to respond, it will remain dwarf, and the reason is that dwarf plant is lacking the enzyme that is going to catalyze the conversion of GA20 to GA1, and therefore what we can conclude now, that the capital LE gene is involved in encoding the enzyme that is GA3 beta hydroxylase converting GA20 to GA1, okay. And therefore if a mutant allele LE is present in a plant, it is going to lag, or it is going to partially express GA3 beta hydroxylase, okay. So I have mentioned an important point, that the dwarf allele, that the dwarf peas we were talking about, it is belonging to Mendel experiment, and therefore we have taken the example of dwarf peas, okay. So we were talking about mutant allele small le, okay. So we also call it as leaky, okay. The mutation is leaky means that the genes can produce a partially active enzyme. Okay, it is having the ability to produce the enzyme, but partially. Okay, and this partial contribution as well as some endogenous GA1 that was already present in the plant gives a little height to the plant. Okay, a plant can whenever we see a plant, it is having some height. Okay, it can be above normal, normal, or below normal. When the condition is below normal, then you will see this condition. Okay, it is having a mutant allele, small alle, which is whose mutation is leaky, as well as some endogenous GA1. Okay, now next thing is that when the condition is more diverse, for example, you can see extreme draft means the height below normal to below. Okay, you can see here, for example, if this is the normal height of the plant, this is below normal, and you can also see a extreme dwarf condition. Okay, now we are talking about this step. Okay, this dwarf mutant is having another allele, that is NA. Okay, and wild type is capital NA. Okay, this allele is involved in the blocking of this pathway. Okay, now we have seen all pathway. It is involved in the blocking of pathway from choline to GA12 aldehyde. And if you remember this step, we have seen here the addition of carboxyl group to carbon 7 as well as the contraction of B ring. Okay, so these allele is involved in blocking this pathway, and if the gibberellins are not going to produce, it will definitely going to produce a dwarf P mutant. Okay, now lastly I have mentioned that homozygous NANA mutant that are completely lacking gibberellins will achieve about 1 centimeter height. Okay, so this will be the extreme dwarf condition, and in the previous slide that I have told you, below normal will be this condition. Okay, and this will be the normal condition, that is having capital LE or a heterozygous condition. Okay, now gibberellin also regulates its own metabolism. As you can see here, endogenous gibberellins generally regulate its own metabolism by either switching on or inhibiting the transcription of genes that encode enzyme of gibberellin biosynthesis and degradation. Okay, let's see it with an example. For example, I have taken a plant and I have applied gibberellin to that plant. Remember, I am talking about a normal plant to which is having a normal height. Okay, and now I have applied a gibberellin externally. Now what will happen? The concentration of gibberellins, for example, the gibberellins that are synthesizing within the cell will stop. Okay, I am repeating, the gibberellins that are synthesizing within the cell will now get stopped. Now the synthesis of gibberellin will get stopped inside the cell. Okay, and as we remember, in the synthesis of gibberellins, the most important enzyme involved was GA3 oxidase as well as GA20 oxidase, and that's why there will be down regulation of biosynthetic genes. Why there will be down regulation? Why these genes are getting off? Because we have applied externally gibberellins, and therefore now there is no need to produce gibberellins to the plants. Okay, on the other hand, there will be elevation in the transcription of degradative gene, that is GA2 oxidase. We have studied that GA2 oxidase is an enzyme that is involved in converting the biologically active gibberellins to their inactive form. Okay, now I have mentioned two important functions of gibberellin. The first one is the hyperelongation of intact stem. You can see here the effect of gibberellic acid on pea seedling. Okay, it is a direct picture. Second thing is that gibberellins can induce rosette plant to bolt by exogenous application. Okay, if you apply gibberellic acids to the rosette plant, more specifically gibberellic acids are GA3. Okay, remember gibberellic acids means they are talking about GA3. Okay, so rosette plants can be induced to bolt, can be induced to flower. Okay, and the question arises, what are rosette? A rosette is an extreme case of dwarfism in which there is absence of internode elongation as well as a compact growth characterized by a closely spaced leaves, as you can see in this picture. Okay, now such rosettes can be induced to bolt. Okay, you can see here in the picture, hyperelongation of stems in rosette plant is invariably brought about by the application of small amount of gibberellin. Okay, you can see here unflowered rosette is now flowered, okay, by application of exogenous gibberellins. Okay, similarly you can see here in the case of Brassica napus. So lastly, this is I have enlisted the overall functions of gibberellins. So first one is the elongation in stem length, second one is inducing internodal growth, bolting as well as vernalization, pollen germination, parthenocopy, it can induce parthenocopy, and you can increase the fruit size of apple by application of GA4 and 7. It is involved in delaying senescence, delaying aging of any plant, as well as it is more famous for malt production, for which generally GA3 is used. Okay, so this was all about gibberellins. In the next video tutorial I am going to talk about the signaling pathway of gibberellins. Okay, so if you understood this video, if you get knowledge, at least a little knowledge from this video, please like, share, and also subscribe to my channel. My channel is entirely based on life science topic, more specifically including biochemistry topic, practicals, etc. Okay, and if you have anything more to add, any comments or any kind of any kind of suggestions you have, you can definitely mention in the comment box. Thank you so much for watching this video.